Hello, everyone. Yes, so my name's Phil. I work in the University of Manchester, and I'm also going to promote the Elixir UK stall. But I don't think I can get a beer as well, isn't it? Can I? Maybe it's worth a try. Okay, I'm going to talk to you today about our crate, particularly capturing fair research outputs in bioinformatics and beyond. I'll just make that a bit larger. Okay, so first a question, a very important question. What is RO Crate? I'm going to read this so I get it correct. An open, community-driven, lightweight approach to packaging research objects with their metadata. So I could say, why would you be interested? Because using RO Crate makes research outputs more attributable, interconnected, and fair. And we quite like this diagram of the conveyor belt of RO Crate to enable packaging research data with their metadata. And when we do research, we generate lots of outputs like data, workflows, publications, and so on. And these can be published in different places. RO Crate is a way to interconnect these different places and pieces of research together to make your research more executable, discoverable, and reproducible using the power of linked data and RDF. And it's all about provenance, capturing the why, the what, the how, and the where of your research. Many benefits of using RO Crate. I'll mention some of these here. It uses linked data, which means RO Crate metadata is naturally interconnected and interoperable and queryable. The specification is general enough, so it can be used in many different infrastructures and domains to describe all kinds of research objects. There are custom RO Crate profiles, which I'll mention a few of as we go on through this talk, that extend the specification to meet specific needs in particular domains and communities. And it enables credit and attribution for all contributors, including ones in typically underrepresented groups, such as people gathering samples or processing sample distribution. And here's some examples of RO Crates in practice across bioinformatics. So with Elixir, the European Life Sciences Research Infrastructure I mentioned before, and we have multiple projects that publish pipeline executions as workflow run crates. And that is one of those extended profiles I mentioned of RO Crates. That captures the provenance, such as who ran the execution, its inputs, outputs, and the execution environment. And an example here of a project is BioDT, which stands for Biodiversity Digital Twin, which was an EU project to develop prototypes working with digital twins and simulations to study biodiversity dynamics. And most closely, we worked on wild crop relatives, capturing the workflow executions as workflow run crates, which are then published on Destination Earth database data lake. And I mentioned here as well, Galaxy. Can I have a word for Galaxy as well? Wendy's not here? Oh, okay. Yes, so you can publish WiFX and Galaxy in other EU projects. These can publish workflow executions as workflow run crates as well. So we're starting to get the picture that it's appearing in lots of places. The Data Plant Consortium, they capture plant science experiments such as in ARC, which is complex objects based on RO Crate as well. So these describe workflows and workflow executions and inputs and results. And then the last one on this slide, Biodiversity Genomics Europe, BGE. This project is defining conventions currently for biodiversity genomics metadata as RO Crate profiles. This is in development. And the new profile aims to capture every stage from sample collection and through to preservation and assembly, genome assembly and analysis. And the COPO platform there mentioned on the slide as well is used to export genomic data as RO Crates. And we use beyond bioinformatics as well with agriculture, language and cultural heritage. And the logo there is for the Australian Language Commons. We've got climate science and material science. And I'll point out as well to other things outside of bioinformatics, particularly with Biofair, which is another stall you should go visit to learn about the UK's next generation of infrastructure for life sciences, biosciences. There's different ways you can use RO Crate in workflows, particularly. We can describe computational workflows registered within the Workflow Hub, which has another poster, which is going to disappear in a few hours, but you can talk to us about that too. And RO Crate export is supported in other workflow engines, including Galaxy, Nextflow, WiFX, and CWL. And we can do data archiving. Long-term data archival is supported, ideal within our crates as well, due to the clearly defined specification and the usage of common web standards. And you don't need to change your data structure. It can work with any type of data, which is one of the great advantages. And it's integrated into platforms including Arrow Hub, FedMseek, which Manaza talked about yesterday, and Paradisic and Dataverse. And trusted research environments. So in TREs, we have a 5 safes Arrow profile, which captures the provenance of workflow executions within TREs, including processes such as sign-off and execution, disclosure control. So the big difference here is that the data, the secure sensitive data, doesn't need to be stored with the metadata, but we have unique identifiers. That means things can be found when they need to be. And we're using the RSA 5 crate profile in projects such as HDR UK, which is another store downstairs. We're quite everywhere, I think, really, at the moment. And EOSC Trust as well. Last slide. Okay, so our crate is open source and global. BOSC is exactly the right place, I think, to talk about open source software for this. And it's open to anyone in our beginner-friendly drop-in sessions. So I've got a few on the slide for the UK and Europe twice a month at 9 and 3 on first Thursday and third Wednesday. Don't expect you to remember that number, but you can get these slides are already on Zenodo, and they will be on. This video is also on our eScience Lab YouTube channel as well. And I'd like to say a big thank you to the people at the bottom, especially Eli, Chadwick, and Stian Solian Reyes, who helped build these slides, and I reused most of them from previous talks as well. So yes, thank you very much.